Welcome back to my channel. Today we're going to discuss about difference between aDNA, bDNA and zDNA. DNA, which is known as the genetic material of us or our life or cell. So there are different types of DNA or different structures of DNA found in our body. bDNA. What is bDNA? bDNA is a predominant form of DNA, and it is predicted that both other forms of DNA are forms from this form under different circumstances depending on several factors like hydration level, DNA sequence, metal ion concentration. So, if the hydration level of the cell or the surrounding where the DNA is present differs, then the structure of DNA will also differ. Then it will form different structures like aDNA or zDNA other than bDNA. So bDNA is a predominant form. When the hydration level changes and the DNA sequence changes, like not the DNA sequence change, like the different types of DNA sequence or in a DNA, there are different parts of DNA, different segments. In few parts, there are purine alternative, the purine repeat sequence. There are purine pyrimidine repeat sequence. There are pyrimidine pyrimidine repeat sequence. Like that kind of different differences are there. So this is DNA sequence. How DNA sequence affect DNA structure. So metal ion concentration, if there is presence of metal ion in the cell or the surroundings. So according to the concentration, how much concentration is present in the cell, bDNA structure can differ. And as the bDNA, that is the predominant form, differ from its original state, it will form different kind of state like aDNA, zDNA. aDNA. It is form of a DNA which was discovered by Rosalind Franklin. As we all know that Rosalind Franklin, by using extracrystallography, she crystallized DNA and gave us a structure of DNA which is not exactly like bDNA. So it was? It was aDNA, because aDNA is formed from bDNA when it is a dehydrated condition, and during crystallization, that is during formation of crystals, there is a dehydrated condition. So the bDNA is formed or is converted to aDNA. So the extracrystallography gave us a structure of aDNA. That is how we discovered or observed aDNA. And what is zDNA, and which promotes zDNA? There are certain factors like high salt concentration in the cell or the surrounding, or presence of 5-methyl deoxycytosine can promote zDNA formation. And also, if there is a region in your DNA which contains purine pyrimidine alternatively in repeating manner, that part is can be converted into zDNA. It's not obvious that it will, but it can be converted into zDNA. Now, what is their comparison? What are their comparison? Why we name them different? Because they have different features. So as we know that DNA is right-handed. We always got to know that DNA is right-handed. Yes, DNA is right-handed because the predominant form of DNA, that is B form of DNA, bDNA, is right-handed. But aDNA, which is a type of bDNA, which is converted bDNA, that is also right-handed. But the zDNA or the zDNA is left-handed. That is, the bDNA is right-handed on this direction, aDNA is right-handed on this direction, but zDNA is left-handed. It's on this direction. So there are different helix ends. So bDNA is right-handed, aDNA is also right-handed, zDNA is left-handed. Now, there are other several comparisons are there. Mean base pair per turn. That means what are the average base pair? What are the mean of the base pair per turn? Per turn means if there are total, if this is a total turn, this is a total turn. So in each turn, in each complete turn, there are 10.4 base pairs. In mean, there are 10.4 base pairs in case of 10.4 base pairs in case of bDNA. And and this is the same, but in egg DNA there is 10.7 base pair. So in bDNA there are 10.4 base pairs, in aDNA there are 10.7 base pairs, and in zDNA it's a little bit high, it's 12 base pairs per turn. Rotation, then comes rotation per base pair. What is rotation per base pair? Now, what is rotation? This is one base pair, this is one base pair, this is another base pair. So if there is a rotation, right, there is a rotation, as you can see there's a curve. So this rotation angle, this is rotation per base pair. This is for bDNA is 34.3 degree, 33.6 degree, and zDNA is almost double. It's a lot. It's 60 degree. Repeating unit. Repeating unit means if in a DNA there is a repetition of purine pyrimidine bases. So in bDNA there is one base pair repeat. In aDNA there is again one base pair repeat, but in zDNA there is two base pair repeat. As we have mentioned before also, that zDNA can be formed when purine pyrimidine alternatively get repeat in repeated manner present. So there is a repetition of two base pair, right? So this is for zDNA it is two base pair. For the bDNA and aDNA there is one base pair only. Rise per base pair along axis. That means what is rise? If this is the, wait a minute, let me remove this. If this is the axis, if this is the axis, okay, this is the vertical axis, then the rise between two base pairs, this is called rise. This is called rise. So if the rise between two base pairs is different for different DNA structure, for vDNA, aDNA, and zDNA, it's different. How much different it is? For vDNA, it's 3.3 angstrom. For aDNA, it's 2.3 angstrom. For zDNA, it's 3.8 angstrom. And diameter. What is diameter? Now, let's take another color. What is diameter? From this to this area, the width of DNA, this is diameter. This is 20 angstrom for vDNA, that is in the predominant form. Then what's the difference in A and Z DNA? For aDNA, it's a little higher, that is little, the diameter is little higher, it's 23 angstrom, and zDNA, it's 18 angstrom. It's a little lower than bDNA. And now there is a base pair tilt, the base pair tilt and propeller twist. Those also show difference. And what are base pair tilt and mean propeller twist? As you can see in this diagram, that there are different axes, x, y, and z. And in x it's a shift, for z it's a rise, and for t or for a y it's a slide, and for t this is tilt. So what, x y z axis, there it is, all the axes are based on the based on the vertical helix axis, or on the basis of vertical helix axis these axes are drawn. And these are the nitrogen bases, these are the hydrogen bonds, these are the hydrogen bonds. So what is tilt? Just focus on this picture. What is tilt? Tilt is a shift of short axis of base pair relative to vertical helix axis. So this is the shift on the shift of short axis. So from this should be like this, the base pair should have been like this, but it is shifted towards this side. So this is called tilt. So now the base pairs are present like this. Now why this base pair shift or make the tilt? Just to make the DNA stabilized, because we make we have to make the DNA well stabilized so that it doesn't denature easily. So to make it more stable, the most stable condition, this state occur. And roll, what is roll? It is the shift of a long axis base pair relative to vertical helix axis. So this is the vertical helix axis, and this is this is its rotation angle. So now this base pair should have been like this, should have been like this. This is the base pair that should have been, but it gets rolled to this one, the upper one. So this is called the roll. And what is twist? It is the twist of bases of base pairs against each other, just like a just like a plate. It just twists itself on this anti-clockwise manner to make it stable, to make the DNA stable and make the nitrogen bases or hydrogen bonding stable, so that that configuration become very very stable and don't denature very easily. So as the DNA form different structures, so different structures means there are different, as we saw before, that there are different mean base pair pattern, there are different rotation, different repeating unit. So we also need to make different tilt or different propeller twist to maintain the balance, right? So in BDNA it is -6 degree. -6 degree means it's on this side, so it's -6 degree. And for ADNA is 20 degree, +20 degree, and for ZDNA is 7 degree. And mean propeller twist, that is this one, mean propeller twist, the twist angle is 16 degree for BDNA, for ADNA it's 18 degree, and ZDNA is 0 degree, that is there is no twist found in ZDNA. There's no twist. So the DNA, so the base pairs will be on the axis, there will be no angle twist. Now, the pitch or turn of helix, that is distance along a 360-degree turn. So, what is pitch? Now, here from this area to this is the region where a helix completes its 360-degree angle, so during this 360-degree angle, how much path it completes, that means the distance. So this is 33.2 angstrom. What is pitch? This region, this total area is 33.2 angstrom. This is 33.2 angstrom. And this is different in ADNA and ZDNA. What is it? In ADNA, it is 24.6 angstrom, it's much less, and for ZDNA is 45.6 angstrom, it is much higher. And then another one is glycosidic bond conformation. So in glycosidic bond conformation, these three type of DNA differs again. So in a BDNA it's anti, in ADNA it's anti, in ZDNA for pyrimidines it's anti, for purines it's syn. Then what is anti and what is syn? So let's focus on these pictures. Okay, so in these pictures, as you can see on the left side, the syn purine. So in purine, the if the CH2OH group and the number one nitrogen are on the same side, then syn means that they are on the same side, and anti means they are in opposite side. So in anti, there is CH2OH group and the number one nitrogen of purine are on different sides, so they are anti. So in syn, they are on number one N1 and CH2H are on same side. In anti, N1 and CH2H are on different sides. In pyrimidines or pyrimidines, there is again N1 present and N1 present CH2OH are on different sides, so they are anti. So this is the difference between anti and syn. So this is how anti and syn nomenclature is given. For purines, purine can be present in both syn and anti configuration, but pyrimidines can only present in anti-configuration. So in ZDNA, purines are in syn configuration, but in BDNA and ADNA, both purines and pyrimidines are in anti-configuration. And now, sugar pakaring. What is sugar pakaring? We all know that DNA pentose sugar. There is presence of DNA pentose sugar, and this is the generalized structure. But in DNA, it form a chair form to stabilize itself. During formation of chair, so there are changes in structures. What are changes that may make? There are C1 prime, C2 prime, C3 prime, C4 prime, and C5 prime, because it's a pentose sugar. So the C2 carbon then goes into the body. If the C2 goes into the, if we draw, this is a 3D structure. See, if we if we observe the 3D structure, the C2 carbon goes into the wood, and C3, that is in opposite plane, and C3 comes out of the board. So if this happens, this is called C2 endo and C3 exo. And for in and if the another thing happen, that is the opposite thing happen, the C3 goes into the board and C2 comes out of the board, then it is called C3 endo and C2 exo. So these are the structures. So how these are made? So these are these are made because to stabilize the pentose sugar structure. So these are the chair form structure, and these are made for to stabilize these structures. So C2 endo and C3 exo, C3 endo and C2 exo. These are the forms that can present. So this can also be found in C2 endo and C3 endo form. So these are the different kind of forms. In BDNA, we can find C2 endo form, that is this one. And in ADNA, we can find C3 endo form, that is this one. And in ZDNA, we can find both C2 endo and C3 endo. And in major groove, and now major groove and minor groove. What is major groove and minor groove? In simple way, we can see the direction or the side where the N7 is of purines are present, that side is termed major groove. As here there is N7 of purine, and there is also N7 of purine. So this can be termed as major groove. And the side where N3 or nitrogen 3 is present of purine, this can be termed as minor groove. But there are characters, there are few characters of major groove and minor groove. Major groove. Major groove in BDNA is wide and deep. In ADNA it's narrow and deep. In ZDNA it's flat. There are no different structure in ZDNA, differentiating structure. In minor groove, in BDNA, narrow and deep. In ADNA, is wide and shallow. And in ZDNA, it's narrow and deep. In ZDNA, there are no differentiated structure in major groove, but there are the narrow and deep minor groove present in case of ZDNA. Thank you so much. This is what today. Please like, share, and subscribe.